Alright, just for fun, today's lecture is brought to you guys from outside. Been on a golf course enjoying the day, so figured why not. So some things to talk about is that life requires energy. So almost all ecosystems, energy ultimately comes from the sun. This is one of those key points. Here's the golf course action. I don't know if you guys could hear that. Somebody's yelling in the background. But almost all ecosystems, energy comes from the sun. That's one of those kind of continuing themes that we talk about. The energy ultimately comes from the sun. So in photosynthesis, some of the energy in sunlight is captured by chloroplast. Okay? Atoms of carbon dioxide and water will be rearranged. You guys have had chemistry, you know, nothing's created or destroyed. We're just going to rearrange it. And we'll rearrange carbon dioxide and water to produce sugar and oxygen. So that's our theme here. In cellular respiration, so this would be the next lecture that we're going to talk about. We'll take the sugar, we'll break it down into water or carbon dioxide and water. So we'll look at that. But the cell captures some of the energy released to make ATP. So that's going to be our little energy molecule. Energy. Alright, so cellular respiration takes place in the mitochondria of eukaryotic cells, and in these energy conversions, some energy is lost as heat. So we can lose energy, but we don't lose the molecules. Okay, so the basic thing here is we're going to focus on photosynthesis today, and notice CO2 and water will go into the chloroplast. We'll get the energy from sunlight. That's what's happening here. And from that, it'll go through this process that we'll talk about today, and we'll get organic molecules. Usually we refer to glucose. Okay, and we get oxygen. Later we're going to talk about it, and we'll revisit this before we do cellular respiration, but in cellular respiration we'll take those organic molecules in the oxygen and put them into the mitochondria. We'll generate a whole bunch of ATP and energy, which can power our work. We'll lose some heat energy, but our products end up being CO2 and oxygen. So this whole thing is a nice cycle that works together. So that kind of introduces what we're talking about. We're going to try to cover as much of this stuff as possible, all three of these things. Maybe not so much the global significance right now, but we're going to try to do as much of this chapter as possible. Okay, so an introduction to photosynthesis, and there's a lot. We do need an essential question, which when I'm looking at this does not look like it comes out properly. Let's choose a different pen color. That's a little better. So our essential question is, what is the process of photosynthesis? It's a long one today. So what is the process of photosynthesis? And how does it convert? I'll say light energy to chemical energy. Something along those lines. So it's a long one, and remember I'm sitting outside and there's lots of sunlight, so I'm having trouble seeing my own screen, but we'll make it through, I think. So some of the kind of big picture stuff we need to talk about are that plants are autotrophs. So they sustain themselves. The actual kind of prefix auto means self. Oops, let's see. Self. Okay. And the suffix trophs means nourishment. I'm writing all over the screen, but basically plants are self-nourishers, so it's self-nourishment. That's how they're able to sustain themselves. They don't usually consume organic molecules derived from other organisms, only from themselves, so do not get things from other organisms. They can get it from themselves. And they make their own food through the process of photosynthesis. So this is their own food making process, okay, in which they convert CO2 and water to sugars and other organic molecules. That's our big picture thing. So photoautotrophs use energy of light. So photo means light. So maybe get some wind out here today. So photo means light. Auto is still self, and troph is nourishment. So we're getting nourishment to ourselves from light. Okay, so we use the energy of light to produce organic molecules. Chemoautotrophs, so they would be using chemicals to self-nourish, are prokaryotes that have inorganic chemicals, so things that don't contain carbon. So it contains carbon, nope, okay, as their energy source. Heterotrophs, hetero means different, this would be us. So it's different is what hetero mean, and troph is still nourishment. So we have different nourishments. So they have to consume or feed on plants or animals or decompose organic matter. So this is getting nourishment from different places, different nourishment. Okay, so photoautotrophs are light self-nourishers. Feed us, they clothe us, we get cotton. They house us, there's wood. They provide energy for warmth, light, transport, and manufacturing. We burn wood, that's us getting kind of both warmth and light. And we can help transport around by using kind of wood and other things as vehicles. Manufacturing, so all that stuff. All right, so some different things that we have. These are different types of photoautotrophs. There's regular plants. These guys, the kelp is actually a protist that we'll talk about a little bit more later on. Probably be next semester. And these guys are some kind of cyanobacterias that actually will photoautotrophs. They're kind of are photoautotrophs as well. So photosynthesis in plants takes place in the chloroplast, and that's a big one to know. Okay, photosynthetic bacteria, so some of those guys have enfolded regions of the plasma membrane containing clusters of pigments and enzymes. We're going to look at all the pigments and enzymes today, and they're going, if you could think about that, those enfolds there will be very similar to some of the stuff that we find in the chloroplast. Chlorophyll is an important light-absorbing pigment in chloroplast. It's responsible for the green color of plants. There's a whole thing to it, but we'll leave that be. And it plays a central role in converting solar energy into chemical energy. So that's what we're talking about in our kind of essential question, is we're taking the light energy and making chemical energy. So chloroplasts are concentrated in cells of the mesophyll. That's the green tissue in the interior of the leaf. Okay. We have stomata. These are tiny pores in the leaf that allow carbon dioxide to enter and oxygen to exit. So we have kind of, through our nose and our mouth, we're able to breathe. That's our kind of opening for that, for gas exchange. For plants that don't have that, they're just going to have pores in the leaf that allow for that gas exchange of carbon dioxide and oxygen. There are veins in the leaf that can deliver water. For us, our veins are for blood and still circulating, but we can have water from plants that help move those things around. So in a leaf cross-section, we'll have these particular cells here. That's what these guys are. These are little cells that all of those little specks in there are chloroplasts. And they'll move around a little bit, but that's where we're getting our kind of energy conversion happening. There's a really cool video that we'll watch that has these guys kind of moving around, called The Dance of the Chloroplast. Chloroplast consists of an envelope of two membranes. So I'll give you the rough version down here. So two membranes, all right, double membrane thing, and encloses an inner compartment filled with a thick fluid called the stroma. So this has its own name of the juicy stuff in the middle of the chloroplast called the stroma. And it contains a system of interconnected sacs called thylakoids. These are little sacs called thylakoids. They sort of look like that. So that one little stack, one little pancake-looking thing is a thylakoid. Okay, thylakoids are often concentrated in stacks. Those stacks are called grana. okay, and have an internal compartment that's called the thylakoid space. Okay, so we'll look at that, but you can think like this little stack that I made in the last slide. The whole thing is called a grana, and within each one is called the thylakoid space. So it's the space within each thylakoid, each little stack. Thylakoid membranes also house much of the machinery that converts light energy to chemical energy. It's got a bunch of the machine stuff there. My dog enjoys being outside too and barking at stuff. Alright, so chlorophyll molecules are built in the thylakoid membrane and capture light energy. Alright, so another way to look at these is sort of the picture that I showed you. There's an inner and an outer membrane. Within those are the thylakoids. They have the thylakoid space on the inside, a stack of them. One is a granum, many are grana. Talk about that a little bit later, and the juicy stuff in the middle is the stroma. Okay. Come on. There we go. Scientists have known since the 1800s that plants produce oxygen. We won't really write this down, but it's a good thing here. But does this oxygen come from the carbon dioxide or water? So for many years it was assumed that oxygen came from CO2 taken into the plant. However, it was later figured out that with a heavy isotope that it comes from the water. So there's this little diagram. Oh, well, they're saying the water there. They just did an experiment and they made a heavy isotope here and a heavy isotope here, and they noted that only when they kind of gave a heavy isotope to the water that they noticed that the oxygen was what they got out of it. They just tracked it along, and so they were able to track it and figure out that the water produces our oxygen. So I'm not going to read through these because I'm going to go through and explain them to you, but if you need it in writing, we're going to talk about two processes or two stages. One of the stages is the light reaction, and so if you need a written version, I would strongly encourage you to come to this one. It's a really kind of generic version that's really nice. The second stage is called the Calvin cycle or the light independent reactions, and this is another good writing kind of written out description of what goes on there. So these are really, really good to look at. Similarly, this is going to be a really good tool to look at here too. So this kind of the series of little diagrams that we'll look at here are really good for getting the overview. We'll have water come into our light reactions along with light. That's why they're called the light or light dependent reactions. We'll have things called NAD plus and ADP come into these. At the end, this is where oxygen is produced. We'll also get ATP and NADPH. NADPH and ATP will go into the Calvin cycle along with CO2, and that's when our ultimate sugar product is produced. And so understanding this diagram at the end of what we go through will be really, really beneficial. That'll be kind of the way to go with it. So it's a lot here. Okay, so we're going to start with talking about the light reactions as best we can. This might be a good place for you to take a break and kind of assess where you're at, and then come back and we'll talk about it some more. Okay, so light behaves as discrete packets of energy called photons. So they've almost got like they don't actually have atoms or whatever. They've got their own little thing and kind of quantity thing that works. It's called a photon. And that's what we call a fixed quantity of light energy. I don't know what it is. I'm not asking you to know. If that's your deal, go do some research on it. Okay, the shorter the wavelength, the greater the energy. Pigments and chloroplasts absorb photons, so that's what's going to happen there, capturing solar energy. It'll increase the potential energy of the pigment's electrons, and it'll send the electrons into an unstable state. We'll talk about that one. Generally, the isolated pigment molecules absorb light. Their excited electrons drop back down and release their excess energy as heat. Okay, so we're going to talk about that. Basically, we'll get a photon of light. It'll come make an electron really, really, really excited is what we call it, but that's just super energetic. Electrons are already energetic enough. They're kind of like if any of you guys work with like kindergarten kids that are maybe a little ADHD, it's like it's already one of those, but now when we get a little light energy, it's also being caffeinated. So it's like your worst nightmare. But it'll reduce as it gets heat and as it kind of burns that caffeine down off and comes down to what we call the ground state. Okay, so within a thylakoid membrane, chlorophyll and other pigments absorb photons and transfer the energy to other pigment molecules. So it's going to be a lot. Let's see if we can get here. We're going to talk about photosystems. So these are light systems that consist of a number of light harvesting complexes that have a reaction center. The complex contains various pigment molecules bound to proteins. Collectively, the light harvesting energy complexes function as kind of a light gathering antenna. They focus it into one spot. And it's really easy. Remember, it's a photosystem because it's a light system. Okay. So these are sort of what the complexes look like. We'll actually have our electron will normally sit here, but we can capture light energy. It'll be transferred to it and will actually excite that particular electron so it's able to do what it needs to do. All right, we'll keep moving. There's a lot to this one. So in light reactions, the light energy is transformed into chemical energy of ATP and NADPH. To accomplish this, electrons are removed from water. That's our big thing, is we're going to split a water and take the electrons there. We're passing from photosystem 2 to photosystem 1. This was a total discovery thing. We'll talk about it later. But at the end, we'll have an NADP plus that will reduce it to NADPH. It's just going to pick up our little electrons at the end. So between the two photosystems, the electrons move down the electron transport chain and provide energy for the synthesis of ATP. This is a really good summary one as well. Okay, so this kind of is a good summary for what we're going to talk about. If you got that, we're good. Okay, the construction analysis is kind of all right. Basically, we'll just have an electron, and it's sort of like, okay. But we add light energy, it excites it and brings it up here so it has greater potential energy. We can move it through and make it do some work as we slide it down to the next part. We need to get it up a little bit higher. We have a photon, excite it so it moves up a little bit again. And when we're done with it, we're going to put it in a little NADPH bucket. So it's actually what happens here, and this might be a good way to look at it for you if you need to. And we can always come back to that and see if it makes sense. So the thylakoid membrane includes numerous copies of two photosystems and an electron transport chain. These are all along the thylakoid membrane. Okay. So the whole thing we're going to look at is in the membrane. Okay. The big thing here to get from, I'm trying to look at this to keep you guys from getting too much. Basically, light energy is absorbed by the photosystems, and they'll excite the electrons. Those electrons will go down the electron transport chain and pump hydrogens into the thylakoid space. Hydrogens are going to then sneak out and they'll produce ATP. That's the basic part. Don't worry about the bottom part at all. This is the main one. I'm trying to get to a picture. Okay. There we go. This is probably the thing to look at. So here's basically where we start. We've got water here, and we're going to split the water and steal the electrons. The electrons will sit right here. Okay, this is kind of like, coming to school in the morning, the electrons are dropped off. They're already hyperactive, but now they get to school, they have a little bit of breakfast, they get a little bit of caffeine, and they're excited. So as we do school, we put them to work. We're going to have these little electrons do some work down this electron transport chain, and the work that we ask them to do is to pump hydrogens across or up their concentration gradient. Look, we've got many more hydrogens here and fewer out here. So we use the energy, the excitement, remember it requires energy to do this, we use the excited energy of these little electrons moving down here to pump the protons into this space. Once we do that, the little electron gets tired. It's tired here at second step at photosystem 1. So we give it a little bit more light. We give it some Coca-Cola at lunch, give it a little caffeine boost, and it's like, I'm ready to go. But at this point, we don't have a whole lot that we can do with it. So it gets to go on a little field trip with a thing called NADPH. NADPH is like an electron chauffeur, When those electrons get done with lunch, it's time to get on the bus and maybe take it to the college. Okay? Maybe it is a little bit more like you guys. Okay? Or our juniors anyway, but they'll get picked up here and that's where they'll go, and they'll go off to do other things. Now, we built this whole thing up here. Notice how many hydrogens we have here. This is kind of like what happens with you guys still too. But put a whole bunch of you in the LGI, it won't take long for some of you guys to go like, can I go to the bathroom? Happens every time, right? So knowing this is going to happen, see, these things are sneaky. It knows these little hydrogens are going to figure out a way out. So as they go like, Miss, can I go to the bathroom? And this little ATP synthase says, that's fine, but can you maybe like take this to the office on your way out? So as these things leave, they are performing a task, which is making ATP. Oh, so, oh, that's fine. You can go to the bathroom. Can you put this together for me real quick? And then it comes together and we have ATP. So that's our general cycle. It's kind of a weird one, but that's basically what happens in the light-dependent reactions. This is light, this is light. It depends on light for this to happen. Okay, so we'll go through it again, but that's the basic kind of setup for that. Photosystem 1, we have our little electron here, it sits here. We're going to energize it with some light. Again, we get those electrons from splitting a water. Okay, the next thing, they go through the electron transport chain, are super energetic and excited. Electrons go through there. They have enough energy to pump hydrogens across their, like up their concentration gradient. Okay, then they get tired. It's lunchtime, so we pump them full of Coca-Cola and food. They get energized again. They're ready to go, and then we put them on a bus, and we're going to ship them over to the Calvin cycle to do some other work. Okay, when we're done with that, we let some of these hydrogens sneak out a little bit. They sneak out. As they do, we ask them to perform a task on the way out. They make us some ATP, and there we go. So the things that we need to go into the Calvin cycle are the NADPH, the ATP, and then we'll get a few other things. Okay, so that's some of the stuff we got there. All right, so that's a tough one, and that's the big one there, but we're going to review that in class. We'll look at diagrams and pictures. If you have questions, you can ask me and shoot me an email, and we'll look at it, but that's the gist of what's happening. The Calvin cycle is a lot more going on, but we're not going to get into too much of it. We're going to do our best to kind of get through the basics here. So the Calvin cycle, or the light-independent reactions, or the Calvin-Benson cycle, or the dark reactions, it has many names, will make a sugar within the chloroplast. So to produce sugar, the necessary ingredients are our CO2 and those ATP and NADPH that we made in the light reactions. So we need a little bit of the energy, and we need those electrons that we kind of chauffeured in this little process. Okay, so the Calvin cycle uses those ingredients to produce this energy-rich molecule that they call a G3P, glyceraldehyde 3-phosphate. I've actually heard it called something else recently, like GAP, or I don't know where it comes from. I think glyceraldehyde phosphate, I guess, is what they're calling it. But G3P works for me. G3P is 3 carbons, and a plant cell uses G3P to produce glucose or some disaccharides or any other molecules they really need. G3P is kind of like a starting point. But it really is, technically, if you want to go with it, G3P, 3 carbons, it's actually half a glucose. Okay, half glucose. That's our ultimate byproduct of this. Okay, press the buttons. Okay, so the Calvin cycle, so there's our ATP that we made in our NADPH with our little electrons. Those are what the eyeball looking things are up top, the little blue parts. Those are electrons. So we need to try to drop those electrons off here. It's a big, nasty process, but we can sort of look at it. Basically, you've got, it's an enzyme called Rubisco, and it helps us take in a CO2 and add it to this thing that we call a PGA. With the use of our ATP and our NADPHs, we can get G3P. Okay, it's not quite exactly what we need, but we can get a G3P. Kind of rearranged there. The G3Ps, notice there's 6 of them that are made every time we put in 3 carbon dioxides. and we'll get one G3P as an output. That will eventually become glucose and other stuff. The other 5 go back in, are used with a little bit more ATP, and we form our kind of starting thing called RUBP. And that is like the super simplified general model for that. I don't typically tend to ask a whole lot other than about reactants and products. So, and that's one of the things that we'll go over in the next class period that we go over this. We'll talk about kind of what goes in and what comes out, because that's going to be the biggest thing that we talk about. Okay, looking at what goes in on the blunt side of the arrow and what comes out at the pointy end. Okay, so that really is the oversimplified version of that. Look at what goes in and what comes out. The thing that we can actually do to this is talk about some evolutionary connections. And there's a really fantastic thing that we'll look at with what was it called? Crash Course, that does a really good job explaining this also. But most plants use CO2 directly from the air and carbon fixation when the enzyme rubisco adds CO2 to RUBP, what we just looked at. Such plants are called C3 plants because the first product of carbon fixation is a 3-carbon compound called 3-PGA. Okay. So that's C3 because that's the first thing that we make. In hot and dry weather, C3 plants close their stomata to reduce water loss, because water loss is a problem. But this prevents the CO2 from actually entering the leaf. So then O2 will build up in the leaf, and rubisco starts adding O2 instead of CO2. So we start really screwing stuff up, because that's called photorespiration, when we start using O2 instead of CO2. It occurs in the light and consumes O2 and releases CO2. So unlike cellular respiration, it uses ATP instead of making it. So it's just burning energy for no reason. You know, it's kind of like I want to get from here to maybe I want to go to Walgreens. You know, it's like I could do it and walk that way. Or I could just run in place, and really that's just burning energy, and I'm not getting anywhere. So that's kind of what photorespiration is. We're just burning the ATP. We're not using it for anything. Ugh, fundamental issues with that system. So an alternate mode of carbon fixation has evolved that minimizes photorespiration and optimizes Calvin cycles. So we have C4 plants because they're named that they fix CO2 into a 4-carbon compound. When the weather's hot and dry, C4 keep their stomata mostly closed and conserve water. There's some other cool things about this, like I think they do, like the light reaction and the Calvin cycle in 2 separate cells. So they'll actually have separate cells, if memory serves me correctly. Okay. So another adaptation in hot and dry environments is called the CAM, and that happens for pineapples, cacti, aloe, and jade. So CAM plants conserve water by opening their stomata and emitting CO2 at night. So they do a day and night thing. One of those processes occurs in the day, one of them occurs at night. That sounds like a good one to do. No, let's just do examples. So here's today's Easter egg for the next quiz. Make sure I would like 2 examples of C3 plants, 2 examples of C4 plants, and 2 examples of CAM plants. Okay. And you're already looking at 4 of them, so I think that you can figure out the other 2. But give me at least 2 examples of our 3 types of plants, and I will give you 5 points on that quiz that we've got coming up soon. So, CAM plants conserve water by opening stomata and emitting CO2 only at night. So they kind of have a day-night system worked out. Separate cells in C4 plants and CAM plants at separate timings, day and night. So CO2 is fixed to a 4-carbon again compound, which banks CO2 and at night releases it to the Calvin cycle during the day. Okay. So, another example there. But basically what's happening here is that the light reactions are happening in the mesophyll cells, okay? And we've actually got stuff going on there, but then in our The Calvin cycle actually goes on in these bundle sheath cells, so they're separated by cells in the two processes. Makes it a little bit better. And then what happens is that we do kind of the light reactions during the nighttime for these guys, and then during the day we utilize the Calvin cycle. So a little different for those. Okay. So I know we've covered an absolute load of stuff, and those two processes of the light dependent and the Calvin cycle probably do not make a whole lot of sense. That's fine. We'll do our best in class to talk about those things and make more sense out of it. But take a look at it. If you have any questions, shoot me an email and we'll get it sorted out.